Great. Thank you so much, and thank you to the organizers for the opportunity to join this amazing meeting and all the foundations that have really come together to put together this amazing program. So my goal in the next 25 or so minutes is really going to be to tell you about one unpublished vignette. And I think we've heard a variety of approaches, starting with some global approaches, moving to some very focused approaches. And I want to tell you, very new work from our group that kind of marries together some of the things that you heard about yesterday in the cancer space together with what we're thinking about in terms of germline variation. But I want to start by contextualizing a little bit about how we think about the world and why we've done what we've done. So each of us is thought to have approximately 100,000 blood or hematopoietic stem cells that reside within our bone marrow, and they're responsible for maintaining and replenishing our blood and immune systems throughout our lifetime. These blood stem cells need to produce every second millions of platelets, red blood cells, as well as a variety of white blood cell subtypes to maintain our health and immunity. And any disruption in this process can result in a variety of diseases. Now, this process occurs through this very elegant and arguably one of the best understood examples of cell differentiation that we know of in physiology, the process of blood and immune cell production, or hematopoiesis, which I've depicted here. And we understand a lot about this process, largely from studies that have happened in elegant and important model systems, including mice and zebrafish, which have and continue to teach us a lot about this process. And yet, when we think about what we see in our patients routinely, as well as just what we generally observe in humans, we often have challenges in mapping those observations onto our current understanding of hematopoiesis. And so we view this not so much as a limitation, but rather as an opportunity to take advantage of observations that we make routinely in patients to be able to better understand aspects of hematopoiesis. And our interest in this really started about 22 or 23 years ago. And at the time, we were interested in trying to understand why the most common monogenic disorders in the world, including sickle cell disease, which is depicted in this electron micrograph, as well as beta thalassemia, which are the most common monogenic disorders, had such variable clinical severity. We knew that there were individuals who would frequently present with extreme clinical symptoms, painful episodes, and other manifestations, and yet other individuals with the same exact mutations would often be relatively asymptomatic. And so that was not well understood. Now, it had been known for decades that increased production of a fetal form of hemoglobin could actually ameliorate the severity of both of these disorders. Now, typically during human gestation, there's the predominant expression of this fetal form of hemoglobin, or gamma globin, shown in the green here. And this then switches off, and there's upregulation shortly after birth of the adult form of beta hemoglobin, which is shown in purple here. And while this switching process had long been known, and it was well understood for decades that this increased persistence of fetal hemoglobin could actually ameliorate severity, how this process was regulated was not well understood. And so as we started to work on this, we failed using a variety of systems. In fact, one of my first projects was actually harkening back to the YACs that were discussed earlier, to do some YAC recombinering and to try to engineer the beta globin locus. All of that failed, and I spent years failing, but fortunately, around that time, some of the first genetic association studies were being performed, and we were fortunate to be involved with Gonzalo Abacassis and Aleida Antonio Cao in performing one of the earliest genome-wide association studies looking at inter-individual variation in fetal hemoglobin. Initially non-anemic individuals, and we were able to replicate these associations in individuals with sickle cell disease, and this revealed 3 regions of the genome associated with inter-individual variation in fetal hemoglobin, including this association on chromosome 2 within the BCL11A gene. And work that we and others were able to do enabled us to show that BCL11A was a key and direct repressor of the gamma globin gene, such that when it's well expressed, which is happening in all of your bone marrow cells right now, there's suppression of gamma globin and predominant expression of the adult hemoglobin. And at the earlier stages of development, there's lower production of BCL11A, and this then favors production of gamma globin due to the lack of repression, favoring production of fetal hemoglobin. And this, of course, not only revealed this important developmental aspect, but importantly also set the stage for lots of translational and clinical work that has now led to the development of therapies that target BCL11A with the idea of actually being able to de-repress fetal hemoglobin and thereby result in amelioration of severity in patients with both sickle cell disease and thalassemia. And so this has now led to FDA-approved therapies such as Exa-cel or Casgevy. But importantly, this also really inspired us as we were starting the work that we've been doing to think more broadly about how genetic variation across the allelic spectrum could impact other aspects of hematopoiesis, and were there opportunities to learn more about this process of blood and immune cell production from this perspective. Now, earlier today, you heard from Amos about the aspect of the fact that individuals can often harbor cancer driver mutations even in the absence of an overt malignancy. And this was one of the earliest demonstrations of this was about 10 years ago when the groups of Ben Ebert, Steve McCarroll, and others reported the fact that there's clonal hematopoiesis in the blood system. And this then, it was thought to be a precursor invariably leading to blood cancer such as myelodysplastic syndrome or acute myeloid leukemia due to clonal evolution of these mutant clones. Now, as Gadi was noting earlier today, of course, since that time we've learned that somatic mosaicism is widespread. Almost all of our tissues have these somatic mosaic clones, but of course this is one of the best understood examples and one that we could readily follow. But in contrast to what we thought 10 years ago, I think we've learned a lot in the intervening period of time, and particularly we've learned, as illustrated in this study from George Vasilio's group, that individuals with these blood cancer driver mutations in the absence of a blood cancer causing clonal hematopoiesis in this case, even with the same exact driver mutation, can have variable rates of clonal growth. So this can vary between an individual not showing any growth or showing regression of a clone over time to individuals showing some degree of clonal growth annually as indicated here. Moreover, the likelihood of an individual with one of these driver mutations progressing to a blood cancer is not invariable and can vary between individuals having no increased risk compared to the general population to individuals having some, albeit not a deterministic progression to a blood cancer. And so this, of course, raises the question, what underlies this considerable variability in individuals progressing? And recent studies have revealed at least some of the causes. We know from a number of observational and interventional studies in humans, as well as a variety of studies in model systems, that chronic inflammation as well as infectious exposures, particularly chronic infectious exposures, can enable some clones to grow out and can also suppress wild-type clones in that setting. And this has particularly been borne out by the fact that IL-1 beta blockade, at least for some types of clonal hematopoiesis, has shown some benefit. We know that exposure to some types of chemical exposures or radiation exposures, particularly chemotherapy and radiotherapy in individuals treated for solid tumors, can often allow certain clones, particularly those with mutations in TP53, PPM1D, and CHEK2 to grow out. But what we wondered was, could there be a contribution from germline genetic variation that's actually enabling, in some individuals, these clones to have a greater growth rate than for others? And so this caught the attention of a medical fellow in our group, Gaurav Agarwal, and Gaurav really wanted to try to take some of the large population data sets that had been generated and also conduct some further studies. And so taking a population with a total of about 600,000 individuals, of which about 40,000 or so of these individuals had clonal hematopoiesis, Gaurav just asked the question, could we identify variants that were either that were found in the germline that were either associated with increased likelihood of detecting clonal hematopoiesis, or could we identify those that are protecting individuals from clonal hematopoiesis? And we specifically wanted to look at the effect sizes of these variants, not necessarily at what their association was. And so by just simply ranking what is the likelihood of getting clonal hematopoiesis or detecting clonal hematopoiesis in these individuals, we could see a number of associations, but we were particularly drawn to this association that was the most protective variant on chromosome 17, or this haplotype on chromosome 17. Now this showed a minor allele frequency in the populations that we were looking at of about 4%. And really we wanted to try to better understand what were the underlying mechanisms by which this was occurring. Now, if we look at this haplotype, which is the sentinel variant of which is marked by this particular variant here on chromosome 17, we can see that individuals with 2 copies of this variant actually had about a 30% reduction in risk of clonal hematopoiesis. In contrast to other variants that had actually been identified previously, this variant protected against all forms of clonal hematopoiesis that we could detect. So this protected against different driver mutations, including DNMT3A, TET2, ASXL1, and JAK2. So irrespective of which of those mutations was present, this seemed to protect individuals. But also, importantly, and in contrast to what had been described for other variants that were actually protecting against clonal hematopoiesis, this also protected individuals from acquiring blood cancers, including acute myeloid leukemia and myeloproliferative neoplasms. And collectively, across all the myeloid malignancies, this variant was protective. And so this particularly caught our attention because we wanted to better understand how was this protecting individuals from both acquiring clonal hematopoiesis and potentially progressing to a blood cancer, and could we understand more about this? Now, beyond that, of course, if we looked at other phenotypes, we saw that this variant was subtly but significantly associated with reductions in all blood counts. These remained in the normal range even in individuals with 2 copies of this variant, but still did cause a significant decrease compared to the general population. And if we look, though, across other phenotypes, across a range of population biobanks, we did not see any other phenotypic association. So outside of the hematopoietic system, outside of a subtle reduction in blood counts, a reduced risk of clonal hematopoiesis, a reduced risk of blood cancers, there was really no phenotypic effect of this variant. So we wanted to better understand what this variant was doing. Now, before I get there, I should just note that this variant is actually widespread. If one looks across different human populations across the world, this variant was present across all of these populations, suggesting an ancient origin, but had been maintained at relatively low frequencies. So its highest frequencies are typically in the 4 to 6% range in European populations, with slightly lower frequencies across other populations, but it's remained relatively low, which might be an issue for discussion. So what is this variant doing, or what is this haplotype doing? So we can perform genetic fine mapping of the potential causal variants, and only a single variant within this haplotype seemed to have a potential functional effect, and this was a variant that was residing within a region of accessible chromatin, as shown here for regions of accessible chromatin in hematopoietic stem cells, and it was also a region that was bound by some key hematopoietic transcription factors, including GATA2, as well as a few other transcription factors that act with GATA2 to enable some of its transcriptional activity. If we look at the accessibility in this region across hematopoiesis, we see that this region is selectively accessible in blood stem cells or hematopoietic stem cells, but then actually becomes inaccessible as these stem cells differentiate. So this really seemed to be a selective blood stem cell-driven enhancer. And importantly, if we also looked at either long-range chromatin interactions or from RNA and attack correlations, we could see that this regulatory region actually interacted or had correlations with the activity of the promoter of MSI2. Within that larger region of association and looking at across about 1 megabase, we can see that MSI2 was actually the only gene expressed in blood stem cells. So no other genes in that region were expressed. If we took single-cell RNA and attack sequencing data from multiomes that we had from the hematopoietic system and looked at a correlation between the degree of accessibility and mRNA expression within the stem cell or early progenitor compartments, we can see a nice correlation between the expression really suggesting that this was indeed a bona fide enhancer. But to directly address that question, we could use CRISPR interference, where we could take a repressive domain, recruit it to that enhancer within blood stem and progenitor cells from humans, and then ask what genes were changed. And indeed, across a number of guide RNAs that we could use that targeted this regulatory element, we could suppress expression of MSI2, but we did not see effects on any other adjacent genes, which I'm not showing you here. Now, there's been an extensive amount of work on this gene, MSI2 or Musashi2, and it's been well studied by a number of labs I'm just listing here over the years for its role both particularly in a variety of cancers, most notably a variety of blood cancers and progression of these blood cancers, as well as a number of stem cell populations. And so this has been well known, but we wanted to try to better understand how is this variant impacting MSI2 function, and how can we better understand some of the mechanisms by which individuals are protected from both clonal hematopoiesis and blood cancers in this context. So to begin to get at this, we really wanted to try to understand what is this variant doing to this regulatory element. And so we could initially conduct some rather reductionist assays. We could take this regulatory element, put it upstream of the endogenous MSI2 promoter to drive a fluorescent reporter, and then simply ask if we introduce this into blood stem cells, what is its activity like? Importantly, we could see that this regulatory element is, when introduced upstream of the promoter, has enhancer-like activity, and that activity is actually reduced by this chip protective resilience variant, as I'll refer to it subsequently. So we saw about a twofold reduction in overall enhancer activity by introducing this specific variant upstream of this reporter. If we looked at this particular variant, it turned out it was disrupting a canonical GATA motif, and as I mentioned earlier, this region was specifically bound by GATA2 within blood stem cells, and so you can see that the G of the GATA of GATA was disrupted. And indeed, if we examine and pull down GATA2 from hematopoietic stem and progenitor cells of heterozygous donors, we could see a reduction in binding of GATA2 to this element. Concomitant with that, if we look at accessible chromatin from hematopoietic stem and progenitor cells of heterozygous donors, we can see that once again there's reduced accessibility of the protective variant, once again showing that this is likely disrupting GATA2 binding, thereby disrupting enhancer activity and chromatin accessibility in this region. Now, we wanted to better understand what are the mechanisms, though, by which this variant is acting, and could we actually model its effects? And unfortunately, we could not precisely recreate this variant using existing base editors or using prime editing approaches, but we could create small microdeletions in this regulatory element of about 100 and 300 base pairs, which we could show in reporter assays behaved identically to the single nucleotide variant. So this gave us confidence that we were at least able to faithfully model that. We could also simultaneously disrupt MSI2 directly for comparison and to examine effect sizes. When we disrupt the regulatory element in blood stem cells, what we see is about a 20 to 30% reduction in MSI2 mRNA expression within these cells. And if we then look at protein expression, we see a similar reduction in overall protein expression by intracellular flow cytometry within this population. And we could get a more profound reduction in MSI2 levels by directly targeting the gene, just for comparison. Now, what was this doing then to stem cells? And so we could initially examine blood stem cell activity in surrogate assays that we have in vitro. And using these kinds of approaches, when we actually either disrupted the enhancer or when we disrupted MSI2 directly, we could see a reduced maintenance of stem cells in these cultures, both in terms of the percentage of cells that are these phenotypic long-term stem cells, either by reducing or targeting the enhancer or by targeting MSI2 directly. And indeed, we could see similar results by just looking at the overall number of stem cells that are maintained in this culture setting. But our gold standard is really to perform long-term xenotransplantation, where we know that we can take these human cells, put them into immunodeficient, and in this case, KIT mutant mouse recipients. That allows their self-renewal and assessment, and then we can wait 4 months for these cells to have long-term engraftment. And if we do that and we transplant an equivalent number of cells into these mice, we actually get a reduced amount of engraftment, as shown here, either by targeting the enhancer, as shown in blue, or by targeting MSI2 directly. This occurs across all lineages, including the stem and progenitor cell compartment, the myeloid cell compartment, the B cell compartment, or the myeloid cell compartment. But interestingly, if we replate these cells, we actually see that there's a skew towards more myeloid colonies. And indeed, if we look at the relative fraction of cells, there's actually more cells that are differentiated towards a myeloid lineage. So I think this is totally consistent with the known role of MSI2 in affecting the symmetric versus asymmetric divisions that blood stem cells can undergo. So this is the loss of MSI2 is really skewing cells towards myeloid differentiation and away from maintenance and symmetric self-renewal. Now, I've shown you so far that we could obviously get at some of the underlying mechanisms, but what was being affected, and how was MSI2 through this enhancer edit actually affecting the phenotype of stem cells? And so we wanted to get at that question, and so we could initially take single-cell RNA sequencing data within molecularly defined stem cells that we could have and look at differentially expressed genes. And we saw a variety of genes that were either downregulated or upregulated upon this perturbation, some of which are likely secondary changes. Most reassuringly, we saw that the most downregulated gene was indeed MSI2, so this was at least consistent with what we would expect. But we could also combine this in collaboration with Michael Karas at Memorial Sloan Kettering, who's thought about MSI2 biology for a long time. We were able to perform this hypertribe approach where we could take an MSI2 fusion with an ADAR editing enzyme. And so this allows us to turn an RNA binding assay that typically is often hard and difficult and turn it into a sequencing problem. So we could just deeply sequence the mRNAs, look for these edits of A to G within the sequencing that we could perform, and thereby determine could there be a potential binding site for MSI2. And indeed, from this, we could actually pull down about a log-fold more potential targets of MSI2 than prior studies had done. We identified about 3,000 mRNAs that were targeted by MSI2. This was mostly bound to the 3' UTR region that MSI2 is known to bind to and was most enriched for the canonical motif to which MSI2 is typically known to be bound to. And so this really was reassuring for us, but we wanted to try to integrate that data together. And if we looked at the impact of those potentially MSI2-bound mRNAs, we saw that on average, these were actually typically downregulated, as shown here in this enrichment plot. And indeed, then we focused on mRNAs that were both downregulated by the enhancer perturbation that I had mentioned, as shown on the y-axis here, but that were also significantly edited, as shown by this hypertribe score. And so this really allowed us to prioritize a group of about 200 mRNAs that were most enriched within this particular network. And this included known targets of MSI2, but also some targets of blood stem cell regulation that had been previously characterized but not well known to be MSI2 targets. So what was this group of mRNAs teaching us, and what could we understand about some of the blood stem cell biology? Well, if we performed ribosome profiling within blood stem cells of normal, healthy donors, we could see that amongst the most highly translated mRNAs in the blood stem cells was actually this group of about 200 mRNAs that are bound by MSI2. So MSI2 really seems to both stabilize some of these mRNAs, but also augment their translation within these blood stem cells. And so we think that this is critical for many of these targets. Importantly, if we look at a variety of the targets of known drivers, such as TEC2 here, in comparing TEC2 mutant cells from the same donor to non-mutant cells from the same exact donor using genotyping methods, we can actually see that the same mRNAs on average are actually upregulated by this drivers. We see similar results for ASXL1, which I'm not showing you here, as well as for DNMT3A. And importantly, if we then look at this group of MSI2 targets that I've shown you, this is actually able to predict outcome in acute myeloid leukemia, as is shown here from the target AML data set, similar to MSI2 expression itself. And so we could stratify both across pediatric and adult cohorts the likelihood of survival based upon the expression of this set of MSI2 targets. But what I've shown you so far has really been focused on how is this variant protecting normal blood stem cells from potentially acquiring somatic mutations. But what about if you already have a somatic mutation? And as I mentioned, this is protecting individuals not only from acquiring detectable clonal hematopoiesis, but also from progressing to or from getting a blood cancer. And so we teamed up with Alex Bick and Yash Prasad's lab in Vanderbilt, and we could actually examine individuals from the BioView cohort that had multiple blood samples performed over a median of about 6 years. And so we could take individuals who had detectable clonal hematopoiesis, that's clones above 2% variant allele frequency. And as you can see, as consistent with prior studies, there was a range of annual growth rates in comparing the first and second time point. But the presence of one of these protective alleles actually reduced that growth rate significantly. This is for individuals with clonal hematopoiesis detected at both time points. But of course, there were some individuals who had clonal hematopoiesis detectable at the first time point, but then that clone disappeared. And it turned out you were about 1.8-fold more likely to have disappearance of that clone just by having one copy of this variant. So this is protecting you and making it more likely that you're going to see disappearance of that variant. So could we model that? Well, we could recreate one of these driver mutations, in this case a mutation, a truncating mutation in ASXL1. We know that that truncating mutation can expand the phenotypic stem cells. This is attenuated by either editing the enhancer of ASXL1 in these human hematopoietic stem cells or by targeting MSI2 directly. We could also see that by colony replating assays, a surrogate measure of self-renewal, that typically ASXL1 is driving self-renewal of these colonies. This could be suppressed by targeting the enhancer of MSI2. And if we perform long-term xenotransplantation into these mice, we can also see suppression of this expansion as shown here, really suggesting that we could get a suppression of this phenotype. We could also perform the reciprocal experiments in collaboration with Michael Karas, and where typically ASXL1 mutant mice show no phenotype. In the presence of increased expression of MSI2, we actually can get an expansion of the stem cell compartment marked by these markers that are lineage negative, SCA1 positive, and KIT positive. And we can also, over several months period, see cytopenias in the mice as well as dysplastic changes consistent with a myelodysplastic-like syndrome in these mice. So I think we can model it both ways. So what I've suggested to you today really is through naturally occurring variation that's protecting individuals and promoting resilience both against clonal hematopoiesis as well as a variety of blood cancers, that we can start to learn about how toggling levels of this key mRNA binding protein, MSI2, could enable protection from clonal hematopoiesis both by limiting the number of stem cells that are targets of these somatic driver mutations, but also once one acquires one of these mutations by reducing the likelihood of that clone expanding, as I've shown you, both in vivo in humans as well as through the perturbations we've performed. And I believe that this is not only teaching us perhaps about some of the interesting biology that might underlie it, but could suggest potential avenues by which we could therapeutically mimic this. And I just take this poster from 1932 from Paris, suggesting that rather than just thinking about treating cancer, perhaps in the future we can actually think about preventing cancer, especially in individuals at high risk. And these types of pathways mimicking nature could help us. So with that, I hope I've suggested at least some of the work that we've done and given you one vignette on how we can take advantage of human variation to try to better understand aspects of hematopoiesis and hopefully uncover some new aspects of that biology. In just finishing, I wanted to acknowledge Gaurav Agarwal, who's really led this work in close collaboration with Michael Karas, our funding sources, and I want to thank you all very much. Thank you.